Okay, hello everyone again. We are continuing the Mitosis Part 1 PowerPoint, which I have posted, talks pretty much about the process of the cell cycle and the primarily interphase being G1, S, G2, what chromosomes are, how they're arranged. What we're going to talk about in this one, which is Mitosis Part 2, is the actual M phase where the cell actually divides. And there are stages involved. You do kind of have to know a summary of what happens in each stage. That's what we're going to go through. So the M phase has two parts. One is mitosis, when the nucleus divides and makes two nuclei. And the other part is cytokinesis, when the cell actually splits into two cells. So at the end of the M phase, you have two new cells that are identical. They each have an identical nucleus. And again, this happens in all cells except for sperm and egg cells. So those are somatic cells. So somatic cells are all body cells except for sperm and egg cells. So it happens every place except in the production of sperm and egg cells. So all your growth from the time you're one cell to your current size was done by mitosis. And then in addition, replacing and repairing tissues is also done by mitosis. All the growth of an embryo is mitosis. So mitosis has 4 stages. And one way you can kind of remember them is using PMAT. So P stands for prophase, M for metaphase, A for anaphase, and T for telophase. We're going to go through what happens in each of these 4 stages, beginning with the first one, which is prophase. So prophase is the first stage of mitosis. During this time, the chromosomes condense into that X form that you can actually see with a microscope. Before that, they're kind of that bowl of spaghetti, and they're not really visible as individual chromosomes. Each chromosome has its sister chromatid attached and at the centromere. The other thing that happens during prophase, I'm going to show you a picture in a minute, is that the nuclear envelope disintegrates so that the chromosomes are no longer contained inside of the nucleus. And then the third thing is something called a spindle, which are really these like protein fibers assemble. This YouTube might work if you click on it on your computer. It's a YouTube video. So I'm going to go to a picture here. So this is the cell before it enters into prophase. So at that point, the chromosomes are not in their little X shape. There's a nuclear envelope, and there's no spindle, which we're going to talk about in a minute. So during prophase, the 3 things that happen is that the chromosomes form the X shape with the sister chromatids attached. So again, in a human cell, there would be 46 of these inside of the cell. Then the other thing is this nuclear envelope disintegrates. So you can see in this picture, there is no nuclear envelope. And then the third thing that happens is the spindle is forming. And that's what these yellow things are here. They move to each end of the cell, and then they send out these fibers that are going to actually attach to the chromosomes in the next phase. So that's prophase. Prophase is chromosomes become visible as these X shapes. The nuclear envelope disappears, and the spindle starts to form. This is a picture in a real cell. It's kind of hard to tell them as much in a real cell because if you think about a human cell, there's actually 46 of those chromosomes, so they're not nicely clear, but this is a cell in before in interphase. And then here is one where you can start to see individual chromosomes forming. Okay, the next phase, or the second phase of mitosis, is metaphase, which is shown here. Meta means middle in Greek, I believe. And in metaphase, the chromosomes line up along the middle of the cell. So this pretend cell only has 4 chromosomes. And so that with its sister chromatids, they're lined up along the middle. And then what happens is that spindle that's being formed started to be formed in prophase, there's a spindle fiber from the spindle that attaches to each chromosome at the centromere. So remember, the centromere is where the sister chromatids are held together. So you can see that there's one attached to each side. And that's basically what happens in metaphase. The chromosomes line up in the middle, and the spindle fibers attach to the sister chromatid. And this is a picture of a real cell. So again, here's chromosomes there, but here you can kind of see that they're lined up in the middle of the cell here. Again, when you have lots of chromosomes, it's actually a little bit harder to see. Anaphase is the third phase. And anaphase, those spindle fibers that were attached to each side of the sister chromatids, they get shorter. And so when they do that, they're pulling apart the sister chromatids. And remember that the sister chromatids are identical copies. So you have one copy going to this side, one copy going to this side for each of your chromosomes. So this is a way that each half of the cell is going to now have a complete identical set of chromosomes, because each of them are moving apart to opposite sides of the cell, and so they're identical ones. That's pretty much all that happens in anaphase, is that the sister chromatids get pulled apart. Once they're pulled apart, they're no longer sister chromatids, but full-fledged chromosomes. Here's a picture of a real cell. This was metaphase, where the chromosomes were lined up, and here you can see them pulling apart. That's probably the easiest one to actually see in a real cell, but you can see one set moving to this side, another set moving to that side. The last stage of mitosis is telophase, and telophase is kind of the opposite of prophase. So in telophase, the chromosomes, you now have one set in one half of the cell and another set in the other half of the cell. So what happens is the spindle falls apart because you don't need it anymore. The nuclear envelope reforms. So you have a new nucleus again, but you have 2 of them now, one in each side of the cell. And then the chromosomes are going to go back to that bowl of spaghetti thing where they don't look like the X-shaped. So those are the 3 things that happen in telophase. The spindle falls apart. The nuclear envelope reforms. And the third thing is that the chromosomes decondense, or they're not the little X-shaped anymore. Can't really fit all that there. So you now have 2 identical nuclei inside of the cell. So the final thing that happens is cytokinesis. Cytokinesis is that cell dividing into 2 cells, which is kind of what you can see happening here. That happens simultaneous with telophase. Technically, mitosis is the division of the nucleus. So we've completed division of the nucleus. We now have 2 nuclei inside of the cell, but what we have left to do is to divide the cell into 2 cells. And so that's what's happening here. is you can see you're going to now have 2 new cells forming, each with a nucleus. And here you can see it. These are pictures of real cells pulling apart to form 2 daughter cells. So that's the process of cytokinesis. That's the actual splitting of the one cell into 2 cells, each of which has a complete nuclei. So those 2 new cells are called daughter cells. And the original cell is the parent cell. And even though they're always called daughter cells, there are no son cells. This for all of your growth. So from the time you're a fertilized egg until you become an adult, all of that growth is by mitosis. When you heal a cut or a broken bone, damaged tissue, or when you replace cells that lifespan is shorter, like your bone cell, blood cells, skin cells, etc. So that's the end of the discussion actually of cell division. I'm going to talk a little bit about cancer now, because basically what cancer is uncontrolled cell growth. So normally your cells only enter the mitosis phase when they get a signal that you need more of those cells. So what happens in cancer is that that process is abnormal and cells are reproducing when they shouldn't be, so that the normal controls on cell division are missing. There are 2 different kinds of tumors. I'll show you a picture in a minute. Benign tumors are not considered cancerous because they don't spread. They can still be dangerous. Like if you have a big benign tumor in your brain, because your brain is inside of your skull and there's no extra room there, it can still impair brain function, but it's not going to spread to other parts of your body. Whereas malignant tumors can spread to neighboring tissues and even to distant places, like a completely other tissue. So you could have breast cancer and it can spread to your brain. But all of these types of cancers result from abnormalities in our genes that normally control the cell cycle. So normally we only turn on that cell leaving G1 and going to S, G2, and mitosis when we need more of that cell type. And if that control gets messed up, then we can be making cells when we don't really need them, and that's what forms a tumor and the development of cancer. Usually that occurs slowly, and usually there's more than one kind of thing going wrong that has to be happening at the same time to actually get a full-blown tumor. So this would be an example of a benign tumor. So a benign tumor does not spread. So say this is an epithelial cell. Epithelial cells are cells lining things like your skin is epithelial cells. And we have one cell here that has some abnormality that causes it to begin dividing when it shouldn't be dividing. And that can continue and form this tumor. But this tumor is what's called encapsulated. That means it's inside the skin, surrounded by normal cells. So it's not really spreading from that spot in the epithelial tissue. So that's why it would be considered a benign tumor, because it doesn't have the ability to invade other tissues or organs. This would be an example of excuse me, where a cancer is actually spreading. So here we have our normal epithelial tissue again, and here we have that one abnormal cell that we were showing before. And usually there are a multiple number of things that have to go down. So here we have this tumor starting to grow. In most cases, your immune system will detect that there's something wrong here and get rid of that cell. But under some conditions, one of the other mutations could be that whatever that repair system is not working right. So you go on to form this tumor, which is these abnormally dividing cells. And then an additional change occurs that these cells are able to break out of that original encapsulated and spread either into other tissues. And this can either also occur in the blood or in the lymphatic system, so they can establish secondary tumors at another site away from the original tumor. So when a cancer spreads, it's because some of these cancer cells from an original tumor were able to get to another part of the body and establish another tumor there. These are just not pleasant pictures of tumors. This is a breast cancer tumor, and this is a colon cancer tumor. So cancer cells are different than normal cells. And one is that mitosis is uncontrolled. And one of the differences is that the cells lack something called contact inhibition. And contact inhibition is when you grow cells, whether they can be normal cell, human cells, in vitro, which means out of the body, like in a petri dish or some type of flask. And so here's your cells, and they will divide until they fill up their space. And it seems that when they are all contacting each other, that's like a sign to them that we filled up our space, we stop dividing now. If you take cancer cells and do the same thing with them, they don't stop dividing. They keep dividing and dividing and dividing. Even though they filled up their space, they just keep making layers of tumor cells. And so that's what the loss of contact inhibition means, is that they don't say, okay, we don't need any more of these cells. They just keep dividing without any control over their division. And that's what leads to the tumor formation. So that's the contact inhibition. The cells also, because they're dividing so quickly, just become different characteristics than the normal cells. They can have abnormal nuclei because they don't completely go through a normal mitosis. And they form these multiple layers. And so they're just have lots of different characteristics than a normal controlled dividing cell. This is a really interesting book. And usually if we were in class, I would talk a little bit more about that. This is a book called The Immortal Life of Henrietta Lacks. It was also an HBO movie, which takes a short part of the book. I haven't actually seen the movie because I don't have HBO, but Oprah Winfrey is in the movie. That's why her picture is here. This is the real Henrietta Lacks. And Henrietta Lacks, this is a very readable book if you like to read. It's a good book to read. Was a woman who lived in Baltimore in the 1950s. She was in her 40s. And she developed cancer and had gone to Johns Hopkins. She was poor. Johns Hopkins had a free clinic for people who couldn't afford health care. And she had her cervix removed. Excuse me, not her cervix, her uterus removed for uterine cancer. And at the time, they had never been able to get human cells to grow outside of the body. So there was a lab that was at Johns Hopkins, a research lab, that whenever they removed a tumor from a patient, they would give a small piece of that tumor to this lab, and they would try to get it to grow in vitro. That means in like a petri dish rather than inside of a body, because they wanted to study the characteristics of cancer cells and what made them so different. And Henrietta's cells was the first time they were ever able to get cells to grow outside of the human body. They're called HeLa cells. They still use them today. HeLa stands for Henrietta Lacks, so they were named after her. They still are used today in research. There's been thousands of experiments done with HeLa cells. If you Google HeLa cells, you'll get, millions of articles come up about HeLa cells. The book is interesting because it talks about kind of the personal implications. Henrietta, although she did get the surgery and some radiation treatment, but she did not survive long after the surgery. And no one ever knew that her cells were the first in the family, first that were able to be grown. And Johns Hopkins at first would give them to other researchers when they were interested in doing research on cancer cells, but eventually it became such a big business that actually there were companies developed to produce and sell these cells to researchers. Henrietta's family were not aware of this and actually did not get any money from that process. And so that's kind of a lot what the book talks about, is both the kind of social and moral implications as well as the medical implications of this cell. So it's a really interesting story, and there still are HeLa cells being used in research on cancer today. I was going to mention, this is just one type of cancer. This is skin cancer, which is fairly common here in Arizona because we have so much sun. And skin cancer is due to ultraviolet light damage of skin cells. And there are multiple types of skin cancer. There's basal cell carcinoma, and that's by far the most common, and luckily the least dangerous, because basal cell is not as likely to metastasize. Metastasize is when it spreads to other tissues. So usually if a person has a basal cell carcinoma, it can be removed with a little bit of surrounding tissue. It's not as likely to spread. There's another type called squamous cell, which also is not quite as dangerous, a little bit worse than basal cell. And then the third type is melanoma. And melanoma is in a type of skin cell called a melanocyte. And melanocytes are the skin, or the cell, excuse me, in your skin which produce pigment. So the pigment is produced as a way to kind of protect your cells from sun damage. And so these are like the signs you should look for. So I just wanted to put these up here of changes in particular spots or moles on your skin that you should go for further treatment. One is when a mole or a spot becomes asymmetrical. So it's not like growing evenly, like a circle just getting bigger. Also, if it has an irregular border like this, see how this one is not also like a smooth border, different color within the spot, and if the diameter is greater than 6 millimeters, that's that little ruler like you used in the lab when you measured. So those warrant you to have it checked out, because melanoma is much more serious. It is likely to metastasize and spread to other tissues. And so therefore it can be much more serious. You can have it removed and they'll take some surrounding tissue. You might remember that John McCain had a melanoma on his cheek. And you could see, if you notice, there was like a dent in his cheek where he had had that tumor removed some time ago.